astrobiology seminar. Thanks especially for coming here on Thanksgiving week. Great to see so many people today. It's my pleasure to introduce our speaker today, Professor Randie Bundy from the Oceanography Department here at UW. Randie is interested in astrobiology, so she's coming today to share her research with us so we can learn more about what she's up to. She's been here about 6.5 years. Before that, she was a postdoc at Woods Hole, and she got her PhD at the Scripps Institute. So with that, I'm going to turn it over to Randie to introduce her to her research. So please take it away. I am going to turn off the lights very quickly.
Thank you. Can you all hear me okay? Okay, great. So thank you for all being here this week again on a holiday week. I really appreciate it. And I'm excited to talk to you today about some work that we're doing in the lab related to microbial iron acquisition in hydrothermal vents, which I think is a very fun place in the ocean to work. And I want to just start off by acknowledging all my amazing co-authors here. And since I know we're quite a varied audience and not all of you think about the ocean on a daily basis like we do, I wanted to talk to you a little bit about what iron looks like in seawater. So iron in the ocean is this very kind of trace constituent, but it's very important in seawater. And so to visualize just a little bit what's in seawater in general, if we think about kind of a bottle or a seawater sample, in it we have lots of different dissolved components, including mostly salts, as many of us know, like sodium and chloride, for example. And these are in kind of millimolar concentrations in seawater. And then we also have organics, which are several orders of magnitude lower in concentration, generally about kind of micromolar concentrations. And these can include different degradation products, cellular exudates, metabolites, things that kind of exist inside of cells that might get exuded into the dissolved components of seawater. And then we also can have gases like oxygen, CO2. These are also generally in kind of micromolar concentrations. And finally, we have the trace metals. And this is what our lab group is largely focused on. And these are in much, much lower concentrations, generally nanomolar or even sometimes picomolar concentrations. And a lot of the metals we think about in our lab include iron, but also some other bioactive or biologically important transition metals, such as cobalt, manganese, zinc, copper, nickel, etc. And iron today is very scarce in the ocean, along with these other trace elements, but that wasn't always the case. So if we think back over 3 billion years ago, the ocean was very iron rich. And this was because it was a very low oxygen environment. So iron concentrations were much higher because iron is very soluble at these very low or no oxygen concentrations. However, as we've kind of evolved into this modern, more oxic ocean, the concentrations of iron have dropped precipitously, about 5 orders of magnitude in terms of its kind of free metal ion concentration. And this is because iron, as it's oxidized, becomes much less soluble and it precipitates. And so because of this, many early bacteria and cyanobacteria evolved in this kind of higher iron ocean. And so they've evolved to have metabolisms that are very heavily dependent on iron. And I like this quote from this paper where I pulled this figure that it says, marine biogeochemical cycles and bioinorganic chemistry have co-evolved, and it is preserved in the physiology and genomes of modern descendants of these early cyanobacteria. So because a lot of enzymes are really slow to evolve over time, many of these are very metal intensive still to this day. And so, as I mentioned, in the modern oxic ocean, iron is very poorly soluble. And so if we think about what dissolved iron concentrations might look like in seawater, that's what I have plotted here on the x-axis, in different depth bins on the y-axis down to the deepest part of the ocean of about 5,000 to 6,000 meters. We might imagine that if all the iron in the ocean was only in an inorganic form, its concentrations would look like this green dashed line. So something very low, like 0.1 nanomolar iron. However, this is kind of an average dissolved iron profile in the ocean, binned at these different depths. And so it's a lot higher than what we would expect if the chemical speciation of iron in seawater was only inorganic. And so it's a lot higher than we would expect based on these inorganic solubility limits. And the reason for that is that almost 100% of the dissolved iron in seawater is bound to some kind of organic molecule. And we call these organic compounds ligands. So these are different types of organics that either just kind of happen to be present in seawater or actually purposefully secreted by microbes that can bind iron. It keeps it soluble, it protects it from precipitating. And so even though these ligands kind of enhance the solubility of iron in seawater and keep it at concentrations above its inorganic solubility limits, it's still very, very depleted in seawater compared to what is required by kind of the average microbe. So this plot here is showing the average seawater concentration of different elements compared to the average cellular requirements of different microbes in the ocean. And then the colors are the residence times of those particular elements. And so iron falls to this kind of bottom right corner on this plot, and that means it's very depleted in seawater relative to what's required by kind of the average microbe. And in fact, it falls in this kind of light gray area, which means it's about 100 times more depleted in seawater than what the average bacteria or phytoplankton might need. So it's very, very depleted compared to what's required for growth. And iron requirements vary quite a bit amongst microbes, and chemists or chemical oceanographers, we often depict these iron requirements as iron quotas or metal quotas, which we usually express as an iron to carbon ratio. So that's what's shown in this table here. And you can see that these vary by large groups of organisms, for example, between heterotrophic bacteria, diatoms, and particularly cyanobacteria, as I mentioned, they were evolving in this kind of iron-rich ocean, and so they have much higher iron requirements relative to other, say, diatoms, for example, or other eukaryotes. And heterotrophic bacteria also have quite high iron requirements relative to diatoms, for example. And so some of these iron requirements are based on their evolutionary history and other kind of adaptations that they've made to adjust these metal quotas. So what are some of these kind of adaptations that organisms can have in the ocean to adapt to these new kind of modern oxic low iron environment that the ocean has? Well, they can adjust their kind of metal quota. So this is an example showing, in this case, the iron to phosphorus ratio of different groups, including diatoms in yellow, flagellates in green, and then picoplankton in blue, and then also in different ocean basins that all have different kind of iron distributions. And we can see from this plot that the Southern Ocean, this is our kind of lowest iron ocean basin in the global ocean. Those diatoms and flagellates there tend to have lower iron ratios, so they need less iron. So they've adapted in this kind of low iron environment to basically need less iron. And they can do that in a multitude of ways, either substituting the metal center in different metalloproteins or various other physiological adaptations. And so we can dig into that a little bit more specifically for iron. And this is just one example. So this is a plot of showing different intracellular iron metabolisms within a cell, including kind of iron storage, iron oxidation, et cetera. And it's showing this in different regions throughout the ocean and also groundwater in different metagenomes. And these regions that are depicted here span in a kind of low iron regions of the ocean to higher iron areas. And one thing that I want to draw your attention to is the size of these circles is showing the percentage of total genes in that metagenome that are dedicated to these different metabolic processes. And you can see that in this kind of low iron data set from the surface ocean, we see certain things that kind of jump out. And one of them are these large circles here for siderophore transport and iron transport. And so I want to talk to you a little bit more about what those are. And so these siderophores are one example of a type of organic compound or organic ligand that binds iron in the ocean. And they're specifically made by microbes, and they have a very high affinity for iron, and they're used specifically for iron acquisition. So they are secreted into the marine environment to bind iron 3. So they're exported out of the cell, they complex to oxidize iron 3, and then they are taken back up by the cell through very specific transporters for that particular siderophore. The iron is released, and then it can be used by the cell, and then the siderophore is re-recycled into the environment. And so these compounds can be recycled. They're usually specific to specific microbes, and they also are very specific to the transporter to take them up again. So they've been identified mostly in soil environments, And then we've hypothesized that they might be a mechanism that organisms use in the ocean as well. Another thing that is unique about siderophores and why they're often used by bacteria in soil environments is that they can promote the solubilization of iron from different minerals. So this is showing an iron dissolution rate from dust in the presence of increasing concentrations of one particular type of siderophore called dusvariaxamine B. And we see that in the presence of the siderophore, the dissolution rates of iron are much higher compared to simple reductive dissolution. So in the terrestrial environment, organisms produce these siderophores to solubilize iron from the soil so that they can take it up into the cell. And it's used as a strategy to make more inaccessible forms of iron accessible to the cell. And so we think of these compounds as kind of biomarkers for specific microbes because you need a very specific biosynthesis pathway to produce the compound. But we also think of them as kind of biomarkers for scarce iron conditions because they're energetically costly for a cell to produce. And so they're also produced so specifically for iron acquisition. So we would expect to see them in environments where either iron is in this kind of inaccessible particulate form or it's very low iron conditions. So cells are competing for iron. And it allows us to link these specific compounds to specific microbes based on these biosynthesis genes. And for this community, they can also be pretty indicative of the conditions of the surrounding environment. So for example, if they're present, it usually means that we're in this kind of more like oxygenated environment where iron is likely to be in these more like iron oxyhydroxides or mineral forms or some kind of metal pore environment. So we're interested in kind of probing for the presence of these compounds in seawater, but it's very difficult. The majority of the organic matter in seawater is unknown to us as chemical oceanographers. It's present in kind of micromolar concentrations, but we're in the super salty matrix. There's lots of different organic compounds. It's very difficult to isolate that organic matter and chemically characterize it because mass specs hate putting a bunch of salt into them. So it's kind of a difficult task, and these compounds are kind of a needle in a haystack because they're a very small component of this overall very heterogeneous pool. And so how do we actually characterize them?
Sorry about that.
No, don't worry.
Turn it off.
So we do a few different things. First, we have to pre-concentrate our samples. So we take our salty seawater, we run it across a column and we concentrate the organic matter onto a resin. So this is called a solid phase extraction. And then we elute that material using a solvent so that we can kind of concentrate our sample down to just several microliters from some, 1 to 4 liter sample. So a big concentration factor. And then we inject our sample onto a column using a liquid chromatography system. And we separate out those compounds based on the polarity so that we can introduce them into our mass spec at different times. So we don't just like overwhelm the instrument with all these different compounds. And then we do 2 different mass spectrometry steps. The first is an ICP-MS step. So we connect our liquid chromatography setup to the ICP-MS. And this allows us to get retention times of different metal containing compounds. And then we do the same exact chromatography again, but this time connected to an electrospray ionization mass spectrometer. This is a kind of softer ionization technique. And from here, we can get the mass to charge ratios of the different compounds. So I'll give you an example. So this is an example of what the data looks like from the first ICP-MS step. So what you're seeing here is time or retention time. So this is like over time, our sample coming off the liquid chromatography setup into the mass spec. And then on the y-axis are the counts for iron 56. And so wherever you see a little peak in this chromatogram is a discrete iron containing compound, such as a siderophore. And we can quantify these by integrating the area under this curve, and we know the stoichiometry of iron in the different siderophores. There's 1 iron atom in a given siderophore. So this allows us to quantify them and also get kind of target retention times for our compounds of interest. And we can do this for many metals at once. I also want to use this opportunity to give a little plug for where we do these analyses, which is in the UW Trace Lab, which is in the Ocean Sciences building in oceanography. It's open to all of you. We have many different mass specs there. We also have a laser, and Jacob is the research scientist that is running this facility. So you should come check it out. Okay, our second mass spec step is we connect our liquid chromatography system to this electrospray ionization mass spectrometer. And this is an example of what the data looks like coming off that instrument at just one given retention time. So this is showing the mass to charge ratio of a bunch of different compounds and their intensity. So every little black peak you see here is a different organic compound at just one retention time. So if we had just looked back at this and we took just the retention time where we saw this one peak, we might get hundreds of compounds. And we're trying to find our siderophore in this kind of mess of bunch of compounds. So we use a few different strategies. One is we can look for iron specifically in these different compounds, and we can search for iron in them based on these iron isotope ratios. So we look for compounds that have iron 56 and iron 54 presence in the correct mass difference based on their isotopic composition, and also this kind of anticipated difference in intensity between these two isotopes. So that allows us to kind of like comb through these and figure out which of the compounds could be an iron containing compound. And for this, we use an instrument that is in Anitra Ingall's lab in oceanography on the 5th floor of our building. So we just wheel our LC upstairs to her mass spec, and then Laura Carlson is the research scientist in her lab that helps us with these analyses. So these samples are really difficult to obtain. They're kind of hard to measure. And so we've only recently been measuring siderophores in seawater. This is a map showing different places where we've measured them so far. So the top map is showing samples in yellow that siderophores have been measured in the upper 1,000 meters of the water column. And then the bottom is places where samples deeper than 1,000 meters have been measured thus far. And the red dots are places where we measure kind of total organic matter that can bind iron. So siderophores would be like a subset of that pool. And so most of the measurements have been focused in the upper ocean because that's kind of where chemical oceanographers suspected they might be the most common. That's where there's the most bacteria. That's where the iron is the lowest in seawater. That's where the most action is. So that's why a lot of the work has focused there. It's also just difficult to collect these samples. We have to collect trace metal clean samples from a rusty boat. That's very hard. And when we first started doing these analyses, when I was a postdoc, we needed up to 1,000 liters of water. Now we only need 1 to 4 liters. So our group has focused on kind of expanding these measurements and collecting them also in the deep ocean as well, because that was very underrepresented. And particularly, we're really interested in exploring if microbes are using siderophores in hydrothermal vents. And we think this is an interesting place to look because we have these reduced or oxygen poor fluids that are mixing with oxygenated seawater. We form a lot of these iron particles, so it could be a place where iron is relatively inaccessible to those microbes. And we're also curious if the presence or absence of these compounds in hydrothermal systems gives us insights into different metabolisms happening in this pretty extreme environment. And we think that that could be pretty interesting to think about in ocean worlds, for example. So I'm going to tell you a little bit more about hydrothermal vents and why it's really cool to think about these compounds there. First, as many of you know, we think that maybe the origin of life could have occurred in hydrothermal vents. So they're just really interesting ecosystems to explore. And these extreme environments lead to really novel metabolisms. So as we're thinking about how different organisms might have different metalloproteins or other enzymes, this is a really interesting place to look. So I want to talk a little bit about the chemistry of these water rock reactions at hydrothermal vents. So in this diagram, we have kind of a slice of a typical black smoker type hydrothermal vent. And first, cold seawater kind of percolates into the ocean interior through cracks in the Earth's crust. It starts to heat and it loses some chemical species that the seawater has to these water rock reactions, notably sulfate, calcium, magnesium, for example, but also starts to gain other types of chemical species. And that's as it starts to heat up more. And this is where it picks up a lot of the metals. So we're kind of melting these rocks. So that fluid is starting to gain reduced iron, reduced copper, and some other gases as well, methane and CO2. And so this chemically altered fluid becomes buoyant, it rises, and then the superheated fluid injects into the surrounding seawater. And so previously we thought that hydrothermal fluids might not affect the broader ocean iron chemistry because these vents expel a lot of iron into the surrounding seawater, but this iron, as it mixes with oxygenated seawater, it forms these insoluble iron oxyhydroxides, so it kind of precipitates right next to the vent. And so a lot of this iron is kind of quantitatively lost right next to the vent as sulfides or oxyhydroxides. And that's why a lot of deep sea mining activities are focused around these vent sites. So we kind of thought that no iron in the dissolve phase would kind of go anywhere away from the deep, away from these vents, and that it wouldn't really have a big influence on iron chemistry of the deep sea. However, a few years ago, there was a cruise that was on the Thompson, which is one of our ships here at UW, and it did a transect from Peru to Tahiti and crossed across the southern East Pacific Rise. And they observed this plume of dissolved iron pictured here. So the colors are dissolved iron in seawater from 0 to about 5 to 10 nanomolar, and then from the surface down to 5,000 meters. And this is right where they crossed the hydrothermal ridge axis, and they saw this huge plume of dissolved iron that went, 4,000 kilometers away from the vent. So this was super surprising, and we had never seen this before. And so we're like, okay, there must be some other kind of reactions taking place at these hydrothermal vents that prevents the scavenging of iron right next to the vent itself and allows this dissolved iron to travel long distances. And this has a lot of implications for just microbes in the ocean and for the dispersal of this iron and kind of the overall iron inventory. And so from this data, these authors in this paper hypothesized that there must be some kind of stabilizing agents that kind of protect this iron from precipitation. They hypothesized that these would be organic ligands because that's what we know in seawater protects iron from scavenging. And so they did a few different modeling runs to kind of model how much of these ligands might be coming from hydrothermal fluids. And basically, if they included ligands in their model, they were much better able to model the kind of dispersal of this dissolved iron away from the ridge crest. And so that's how our group kind of got involved with this project, since we study these different organic molecules. And so we did a follow-up study to this region and now to some other vent systems that I'm going to show you. And this work has been led by 3 individuals in my lab. Patrick Monreal, he's an astrobiology student and he's a 2nd year, so he's a new student in the lab. Laura Moore, who I'm going to show a lot of her work as well today, and she just graduated a couple of weeks ago. She's on her way to Antarctica right now. And then Colleen Hoffman, a former postdoc in the lab. So we have so far been looking for these types of ligands in 3 different vent systems: the Mid-Atlantic Ridge, the Mid-Cayman Rise, and then the Southern East Pacific Rise. And these are interesting vent systems to explore because they have very different spreading rates. So the Mid-Cayman Rise is an ultra slow spreading center. The Mid-Atlantic Rise is a relatively slow spreading center. And then the Southern East Pacific Rise is the fastest spreading center that we know about. And the spreading rate is kind of a proxy for how much iron we would expect in these different systems. So ultra slow spreading, we expect there to be very low iron. And then in the fast spreading, we expect there to be quite high iron. So we're looking kind of across a wide range in vent geologies, vent types, spreading rates, and also iron. So first, let's look at the slowest spreading center that we've explored thus far, the Mid-Cayman Rise. And this was work done by Colleen Hoffman and Patrick Monreal. And so this was the first place that we ever tried to look for siderophores in a hydrothermal vent system, and we saw some, so we were very excited. So this is showing you different compounds in the columns. So these are all different siderophores that we identified at least one site at the Mid-Cayman Rise. And then on the y-axis, or these different rows, are different vent sites that we had samples from. And then the colors are the presence, absence of those compounds and kind of our confidence level in the compounds. And some things that were interesting that we found here were, we found some actual similar siderophores that we found in surface waters. And that was kind of surprising to us because there's very different microbial communities present in the bottom of the ocean compared to in the surface. And one of those types of compounds are these siderophores called cinicobactins. And they're kind of interesting because they have these long fatty acid side chains. And so they can actually partition into the cell membrane. And cells can use them as more kind of like a fishing pole. So it's actually tethered to the cell and they kind of like fish for the iron and it doesn't diffuse away from them and then they can pull it back in. So we see those a lot more in the ocean compared to the soil environment. And that makes sense because why would you spend so much energy on a compound that might just like float away from you? However, it was also cool because we saw lots of new siderophores also that had never been seen in the ocean before. So this is just one example here of this Tenaciabactin. So we were pretty excited because we actually weren't sure if we'd see any siderophores at all in these environments. And so the first place we looked, we saw some, but this was also kind of our lowest iron vent system. So then we wanted to move to these other samples from the Mid-Atlantic Ridge, or this slightly faster spreading center. And here, this was work led by Colleen Hoffman. And this particular study, we were able to sample a wider variety of vents. And this was a much broader range in iron concentration. So that's what I'm showing you here. So this is showing dissolved iron concentration across this kind of transect. All the little black dots are places where we have different types of samples. Not all of them do we have siderophore samples, but you can see that here the dissolved iron ranges from near zero in surface waters, about sub a nanomolar iron, to 20 nanomolar in some of the vents that we are sampling. And so the vents themselves have about 2 orders of magnitude higher dissolved iron than surface water. So this was kind of a broader range of iron and also vent geologies, from a rainbow type black smoker vent and then this lost city type vent. And so we also saw a lot of siderophores here as well. In this particular case, I'm showing now the siderophore as rows on the y-axis, and then our different stations where we have siderophore samples on the x-axis in the columns. And again, the colors are just showing the presence or absence of that particular siderophore and our confidence level in them. And then the stations are also kind of organized by iron environment. So low iron, high iron, or slightly off the ridge axis. And some of the things that were interesting from this study were that we actually saw some very similar compounds that we had seen at the Mid-Cayman Rise. And so that was encouraging because these were the first 2 vents that we looked at. And that makes sense that there's also some similar microbial communities among some of the vents in these 2 places. But we also saw this kind of similarly interesting phenomenon where a lot of the siderophores that we identified were these kind of amphiphilic type siderophores, or these types of compounds that would tether to the cell membrane. And so what these 2 plots are showing are kind of our total concentration of siderophores in different samples in picomolar, and then the proportion or the concentration of those siderophores that were these kind of amphiphilic type compounds. And so in some cases up to, 30% or so of the siderophores that we saw were these more kind of amphiphilic type. And again, these are ones that are really common in these very low iron surface waters. And so that wasn't really what we expected, since iron concentrations were so much higher. And there's also very high particulate iron concentrations in these environments. Another thing that was really interesting and unexpected is that actually we saw more siderophores where we saw more iron. So here we have the dissolved iron concentrations on the y-axis, and then the siderophore concentrations on the x. And so in the samples where we had a lot higher iron concentrations, we saw even more siderophores. And that's kind of opposite of what we expected. Actually, we thought that maybe where iron is super scarce, we would see more of these compounds. And that's a little bit what we see in surface waters, but that's not what we saw here. So we also have a little bit of microbial community composition data from this particular study, but only at 2 of the sites. So I'm showing an example from Rainbow and then also from Lucky Strike. And in this Rainbow sample, we have a couple of different size fractions, the 0.2 micron and 3 micron filter. So bacteria present on the 3 micron filter, we interpret that to be kind of particle associated bacteria. And those on the 0.2 micron filter, we think of those as more like free living bacteria. And then the bacteria that I'm showing you here are all only groups that contain known siderophore producers. So the proportion of the total 16S gene copies that we recovered are reported here on the y-axis. So what this means is that up to about 20% of the 16S genes that we recovered were siderophore producing type groups in some of the samples. It was lower in some cases, and up to 20% in, for example, this kind of Rainbow sample. So what that means is that basically siderophore producing microbes were present across these 2 sites, and they were appreciable members of the community in the different sites, but there's lots of differences in the microbial community structure across the different sites. Okay, finally, I want to move to this fast spreading center. So this is the most recent of the work that we've been doing. And this was led by my student, Laura Moore, who recently graduated. And this system is very interesting because we went back to the similar area where that big iron plume was found a few years ago that I showed you in the beginning of the talk. And we primarily focused on sampling this 16 to 18 south sector of the Southern East Pacific Rise, which is known to have a lot of vents. And we went to a lot of different stations that we gave creative names because we were very delirious and tired on the cruise, so they all are kind of funny. But we explored the vents using an AUV, which is AUV Sentry, which is pictured here, and then also water sampling with CTDs, which some of our rosettes are shown here in this photo. And so first, I'm just going to show you the iron data. So on this plot, I have depth, just focused on the deep part of the water column and the depth of where these plumes are, so 2,200 to 2,600 meters. And then dissolved iron concentrations are in the colors. And there's a little gray bar here separating the stations that we did that were just off the ridge axis slightly. And then we have our station names on the bottom here. So in this study, we saw a much larger gradient in dissolved iron than our other two major vent sites. So it ranged from about 1 to 100 nanomolar of iron. The highest concentrations of dissolved iron were generally in the center of the neutrally buoyant plume, like we kind of saw in that big transect before. So that makes sense. And so then we wanted to look at siderophores across all these sites and across this really large gradient in iron as well. And because we know that siderophores are produced by bacteria, we also have bacterial abundance data from this cruise that was provided from Dylan Vecchione in Bob Morris's lab using flow cytometry. So on the top, we have the bacteria abundance, and then on the bottom, the iron data, and then the black box is highlighting the kind of center of that neutrally buoyant plume. So we did see higher bacterial abundances kind of in the center of that plume along with this high iron. And so now if we look at the siderophore data, we have the siderophore concentrations in picomolar in the colors, and we have the same kind of deep depth range that we're focusing on here on the y-axis. And so in this study, we saw pretty high concentrations of siderophores ranging from undetectable to 41 picomolar along this region. And this was higher than we generally see in the surface ocean, where we have a lot higher bacteria abundances and these really low iron conditions where we expect to see these compounds. So it was interesting that we saw such high concentrations in this environment, but the concentrations were also really variable between sites. So we see some kind of hot spots in the neutrally buoyant plume, but you see other places where we also saw high concentrations, like higher in the water column where there was no hydrothermal fluid influence. We also saw some very close to the seafloor where we think there's a lot of these diffuse vents or lower temperature vents as well. So it was very site dependent. And in these samples, we found 8 unique siderophores that we identified with super, super high confidence. So I'm just focusing on those for now. We're very confident in these 8 compounds that we've found because Laura, my student, she visited Florida and she ran her samples on this super, super fancy mass spec pictured here, this 21 Tesla FTICR. So we have very high confidence in our identification of these compounds. And the thing that was interesting about these 8 compounds that she found is that some of them have never been seen before in the ocean and had never been seen in our other vent sites. But then there were also 4 of them that had been found in our other vent sites that we've been exploring. So there's some new things, there's some that we've been now starting to see across several vents. So it seems like there's some amount of compounds that must be common to specific microbes or common iron acquisition strategies across these vents. And we were really excited about this data set because even though this was such a high iron environment, one of the highest that we can see in the ocean, these compounds were essentially ubiquitous throughout the data set. So here what I'm showing is on the x-axis are each of the 8 compounds. And then on the y-axis, these numbers just represent individual samples and it's their sample number. And then they're organized into kind of environments or groups. So on the left here, we have the neutrally buoyant plume. We have samples that were all kind of in background seawater with no hydrothermal fluid influence. We have samples that were found in some of the diffuse or low temperature vents that we sampled. We had a few buoyant plume samples, and then we had some that we sampled that were kind of off of the ridge axis. And basically what you can see is that they don't really look, if you kind of squinted it, it doesn't really look drastically different amongst the sites. So there's these compounds were kind of present everywhere. And the color bars on the side of each are the dissolved iron concentrations, which span this really large range. And there's also not really a pattern with iron environment. So we saw the siderophores present across all these ranges of iron concentrations. And this was also exciting because this data set basically doubled the number of published siderophore analyses that we have in the ocean, because we don't have very many measurements. We saw siderophores across a much wider range in dissolved iron than we expected and that we have seen before. And so it suggests that they might just be a pretty universal iron acquisition strategy in these hydrothermal systems and maybe just in the ocean in general. So we're only starting to look into the microbial community composition data because a lot of this data is still very new. And so this is an example of the microbial community composition, also grouped by environment, provided by Dylan Vecchione and Bob Morris. And so the different colors are different microbial groups that Dylan has annotated. And then I put stars next to the ones that contain known siderophore producers as well. So we do see known siderophore producing microbes across all of these environments. And we're interested in kind of digging into this further, particularly with SEP05, because it was such a prominent member of the community across many of these different sites. And no one has really looked at its kind of iron metabolisms in detail yet. And so Dylan is really interested in that. So stay tuned. Okay, so I wanted to end with a few kind of parting thoughts and summarizing what we've seen in these data sets. So what we found so far is that siderophores are this kind of unique microbial iron acquisition strategy in the ocean. And we are starting to think of them as just kind of biomarkers for a need for iron in this very like oxygenated ocean. So I think previously we thought of mostly the surface ocean as like the only low iron part of the ocean. But I think basically the whole ocean is very low iron for these microbes. I showed you early on that the average iron concentration in the ocean is about 100 times less than what most cells need. And so we seem to be finding siderophores everywhere we look. We also think that the presence of these compounds in hydrothermal vents specifically was a little bit of a surprise given the relatively high iron concentrations, but it seems like they're kind of ubiquitous, and iron might be still relatively scarce in these environments, or a lot of it is in these particulate forms that are kind of locked up in minerals. And so organisms are using these compounds to solubilize that iron and be able to access it and use it for their metabolisms. And the thing that's interesting to this community is that metals are really essential for life. And so strategies that microbes are using to acquire these metals from the environment gives us insights into kind of their metabolisms and what the surrounding conditions are. And some things that I didn't really have time to go into today, and some new things that have been coming out is, for example, different metallophores exist as well besides these siderophores. So there's a lot of other metallophores with different metal centers, and they've been identified for other essential metals like zinc, manganese, nickel, copper. And there's a preprint that just came out where there's a metallophore for lanthanides, and it looks exactly like a siderophore. It looks very similar to rhodopetrobactin, which is a siderophore. It's just a variation of that, but it's meant to bind lanthanides. And so that's a new novel compound that's showing us something about how organisms are trying to get lanthanides. I think that's very exciting. It's also very exciting in this other recent preprint that bacteria that were found at the International Space Station have more metalloproteins than their Earth counterparts. And the authors are suggesting that they have a lot more metalloproteins in this kind of extreme environment to deal with redox stress. So thinking about kind of the metalloproteins and metal requirements of microbes in these very extreme environments and also in ocean worlds, I think is a really important and exciting thing to think about. Okay, with that, I'd just like to acknowledge our fieldwork teams, which were called Plume Raiders and Fridge, and then several other people in many different labs that helped us. And this is our happy group after our almost 60-day expedition to the Southern East Pacific Rise after a lot of COVID drama, but we made it and we're back. And I also want to thank everyone that funded this work as well. Thank you.
Thank you very much, Randy. So we have time for questions.
Yeah.
How do like the searophores work? It's like I'm thinking like hemoglobin, how it, like coordinates the iron.
Yeah.
They work with that.
Yeah, it's pretty similar. So they're kind of these like octahedral complexes. I showed some of them, but they almost look like a little claw. So they're kind of like, you know, the iron is a little bit like sterically hindered in there. But some of them are a little bit different, but that's their general kind of conformation. And it's slightly different than a heme. You know, like they don't have those like foreign rings like that,
They're
but
not peptides though, are they?
no.
Okay.
Yeah. So then, you know, when they bring it into the cell, they generally reduce the iron out of the complex into iron 2, and that's how they can kind of release it because they bind to iron 3 specifically, and then they can recycle it.
Very cool.
Yeah. Yes.
How do the size difference between sedarophores and like vesicles? Sedarophores are much bigger than like a vesicle set, or are they similar?
I think they're kind of similar. I mean, there's been some work done recently with diatoms where they actually like, that's one of the ways that they can like take up a siderophore. They kind of like engulf it like that instead of taking it through a specific transporter. And so that's why I'm trying to think. I don't know that much about vesicles, so I don't know if they're like exactly the same size. Most of the ones that we're looking at range from like 300 to 1800 Daltons or so. Yeah. Yeah, Gio.
I was wondering if you have data whether you see a difference in the, or not necessarily the quantity of sedarophores between the surface environment and the hydrothermal environments, but the proportion of those sedarophores that are free and detached from the cell from one environment to the other, are they generally more free than before?
Yeah, that's a good question. I think we haven't really looked at that yet systematically. But we have more data in the surface, and in the surface in places where iron is particularly low, we see a lot more like free siderophores, or like, do you mean free, like not iron containing?
No, I mean,
Not attached to the cell. Okay, yeah, sorry. That we really don't know. So we've tried looking for siderophores in the particulate phase. So like assuming that, you know, we see these ones that are amphiphilic, so they should be attached to a cell. So they should stick to our filter and we don't really see them, and we don't know if it's an analytical hurdle or not. It could be. And so we haven't really looked at that because we don't have a lot of samples to look at. And when we try to look on the particles, we don't really see them, even though we feel like we should. But it's a little bit tricky because just the types of filters that you have to use is kind of, they're expensive and tricky because you need something that's like trace metal clean, but that you can also put a solvent on. And so we have to use Teflon filters. So it's kind of a pain, but we don't know. That's a good question. Yeah. Yeah.
You know about the temperature and pH stability of siderophores, because I'm wondering where the complexation actually might be happening.
Yeah, that's a really good point. So it varies a little bit, but a lot of them do not bind iron well below like pH 5 or so. Some of them are different, but in general, the ones that we're seeing, that's kind of how they are. Our interpretation is that they're not really produced very close to the vent orifice. You know, we think that that perhaps they're produced more kind of in the neutrally buoyant plume after these fluids have kind of mixed with the surrounding seawater and it's reached like kind of a neutral density. It's, you know, it's still like a slightly warmer fluids, but nothing like these superheated fluids. And then it's very close to the pH of background seawater, which is closer to like 8.2. In the deep ocean, it's probably like about 7.9 or so. So it's still like very much in the range where they're very effective. So that's kind of our interpretation. We don't think that they're coming from the fluids themselves or very close to the vent. Yeah.
You mentioned how sedarophores are a widespread strategy, it seems, in the ocean. Do you know anything beyond the ocean? Are they also widespread? Rivers, lakes, soil?
Yeah, so they're definitely very widespread in soils. And so that's very interesting because iron is obviously super high in soils, but it's all particulate. And so that's why we think that they're producing them there to, again, like solubilize that iron. There hasn't been a lot of direct measurements of siderophores in rivers or groundwater or lakes, but based on genomic information, it appears that the genes to make the siderophores are pretty common across those environments as well, but a little bit less. Like in the one kind of summary plot I showed of just a few metagenomes that were surveyed, there was like fewer percentage of genes were dedicated to siderophore biosynthesis in like groundwater, for example, compared to in the ocean. But groundwater is also pretty high iron and much higher than like rivers and things because it's usually more reduced iron. So that doesn't really surprise me, but otherwise they seem a pretty common strategy in a lot of different environments. Yeah.
Are there any online questions? Okay. Any more questions here?
You're saying like there's dissolved iron, so there's also some dissolved iron in the ocean? Or in what like form is that?
Yeah, so there's basically dissolved and particulate iron in the ocean, and we partition those just like operationally based on a filter, a 0.2 micron filter. So anything that goes through that 0.2 micron filter we call dissolved. And so that's what I was focusing on today. So basically we think almost 100% of the dissolved iron in the ocean is bound to some organic because otherwise it precipitates. And so then all the particulate iron, it's either locked up inside of a cell or it's in an iron oxyhydroxide, or near these vents, it could also be some iron sulfides, for example. But most of the other like particulate iron in the ocean, a lot of it is iron oxyhydroxides that have like autogenically formed because of the oxygenated seawater, or it's inside of cells. Yeah. Yes.
So you mentioned how the transport of siderophores are highly specific to the specific siderophore, but also how those siderophores are highly universal across many different organisms. I think previously maybe there was a thought that this was like a competition strategy to acquire iron, but given that universality of it, is it maybe looking like it might be more on the acquisition side than competition?
Yeah, I think that's really a good question, and I think it's pretty open because we see a lot of organisms that are cheaters, so they can't produce any of the siderophores. They don't have the genes, but they have the transporters for them. So they could just be, you know, like living off this common good. But we're also just seeing like that they might be produced for other reasons. Like there, you know, there could be different symbioses as well, where we see that a little bit in the surface ocean, where like some eukaryotes can't produce siderophores, but an associated bacteria can, and then the eukaryotes providing organic material to the bacteria, and the diatom or whatever can take up the siderophore iron complex. So I think there could be lots of different reasons that they're being produced, and it might not just be competition, could be other things as well. Yeah.
All right, still no.
Yeah.
Question. Connection between organisms that can produce siderophores versus organisms that also like other extreme environments, get a tolerance for repellent environments.
We haven't looked at that yet, but I suspect that there might be a connection. So I think it's going to be interesting to expand this to some other extreme environments. Like we've been talking with Geo about some of the ice environments, for example. Because it does seem like, you know, organisms in these more extreme environments might just like need more metals in general. Like we were kind of seeing, I gave you a little teaser of from like the International Space Station, but we see that in the ocean as well. So it could be that it's kind of a more common strategy in those extreme environments, but we don't know. Yeah. Yeah, good question. Yeah.
So what would the role of the siderophores be like on the ISS? Like, are these just bacteria floating around in the air filters, or?
I don't know, and like they didn't mention siderophores specifically in that paper there, but they are talking about some other metallophores, and so I'm not sure if they tried to look specifically at biosynthesis genes for siderophores, but they do have just like a lot more metalloprotein, so they were kind of like digging into that as to why that would be the case, and their hypothesis was that it was related to dealing with redox stress. And so They didn't look specifically at biosynthesis genes for some of these kind of metallophores that I was talking about, but I think that that would be interesting to look at, because if they have such high iron requirements or metal requirements and they need to get it from somewhere, from their surrounding environment, it's just one of the known strategies that we know of for them to try to acquire it from some inaccessible source.
If you're getting iron, they're like not in a liquid.
I don't know. I don't know. And I don't know where, there was like a few different strains that they were looking at, and I forget where they isolated them exactly from, you know, like inside the, I can't remember if it was like, yeah, from the air filters or scraped off the surface in their lab or whatever, but that's a good question.
All right, well, seeing no more questions, why don't we thank Randie again for a great talk.
Thank you.